For my biology role model project, I chose to do Nina Tandon. So she grew up in New York City, and she always enjoyed doing science experiments when she was little, which involved taking TV parts apart and experimenting with their static electricity. She was always super smart. She took a college-level calc class when she was 14, and her mother encouraged her and her siblings to pursue their STEM interests. So obstacles, she's a woman in STEM, so obviously she faced the pressures of that, as well as she started her own business, so she faced obstacles of being a woman in entrepreneurship. And then her education, she went to Cooper Union for a bachelor's in electrical engineering, and then received a Fulbright scholarship in Rome, where she worked on a device that basically smells lung cancer, which got her interest in health-related path. And then she went to MIT for bioelectrical engineering, and then to Columbia for MBA in health entrepreneurship and a PhD in biomedical engineering, which focused on cardiac tissue. Other hobbies, she likes yoga, working out, rock climbing, metalsmithing, writing, theater, and poetry. And her engagement ring was 3D printed based on the pattern of cardiac tissue, which again was her PhD focus. So I thought that was an interesting fun fact. And then the field of biology that she's in is biomedical engineering. And some questions she's looking at is how can EpiBone successfully grow human bone, organs, tissue, and cartilage in a lab. So she uses stem cells to do this, and she grows it to produce a skeletal structure which is grown outside of the human body and then hopefully be used in skeletal reconstructions. Her research paper kind of focuses on cardiac tissue, which again was her PhD focus. And then her and others created and experimented on a device to transport cardiac tissue and their patches, which cardiac tissue uses patches in transporting. But transporting these patches and the tissue itself while keeping them viable, contamination-free, is challenging. So they created a device to provide the necessary support for tissue growth and maintain viability during this transportation, which also helps in integration with the host after it's implanted. So the figure shows stimulation of these cells. So factors like perfusion and electrical stimulation influence how cells spread in tissue scaffolds, which scaffolds is above. Without perfusion, more cells gather on the outer surface, while electrical stimulation leads to more cells on the outer layer of the cell, which suggests that both perfusion and electrical stimulation affect cell distribution in the scaffolds, keeping them near the surface, which helps in viability of the tissue. And then her company is called EpiBone, which takes the patient's stem cells to produce skeletal structures based on unique DNA profiles, which is able to decrease the likelihood of rejection, simplify surgeries, and possibly shorten recovery time for patients in skeletal reconstruction surgeries. And it's a 3-step process. It takes about 3 weeks, and they've already got FDA approval to use their technology on patients for clinical trials. And then currently it's working on knee cartilage and constructing that. And then that's kind of more in the 3-step process if you want to pause and look at that. And then accomplishments, you can read all those, but as you can see, she has so many accomplishments. She's done so much, and she is so young still. And then the most inspiring things are that, or how it resonates, I admire her most, is her passion for biology and engineering and how she connected those into a path for her, and the amount of success she's able to accomplish, and her ability to be a businesswoman and a scientist in New York City as well as an advocate for diversity and inclusion in STEM. And it resonates with me just because she's very successful in STEM in the medical field, and my dad is an engineer in a healthcare company, which I've always been intrigued in that work. And then I'm really interested in research on stem cells. And then here are some key takeaways for you, and thank you. Here are references as well.